Welcome to our virtual comparative genomics workshop. This is the first in a series of video lectures entitled Predicting Gene Function by Comparative Genomics of Prokaryotes and Eukaryotes. The Institute of Food and Agricultural Sciences, the University of Florida, and the National Science Foundation sponsor our research group to give 2 or 3 day workshops covering this material at the University of Florida and other institutions. IFAS, UF, and NSF have also sponsored the creation of this series of video lectures. The senior personnel giving these lectures are the PI Dr. Andrew Hanson and co-PI Dr. Valérie Decrecy-Lagarde. Dr. Svetlana Gerdes, who also participates in these lectures, is a highly experienced genome annotator, well-versed in metabolism and familiar with the seed. Together, Dr. Hanson, Dr. Decrecy, and Dr. Gerdes created this workshop's material. Dr. Claudia Lerma-Ortiz recorded and edited these lectures and produced some of the PowerPoint material to accompany them. She also assembled all the lecture recordings with the PowerPoint presentations to create these virtual workshop tutorials. This workshop is organized as follows. We are going to cover a number of general topics which Dr. Andrew Hanson will now describe.
First of all, the scale of the unknown gene problem. Just to give you a feel for how many unknown genes there really are out there. Then to talk about how eukaryotes, more specifically, but not solely plants, and eukaryotes share many genes. Then we'll talk about comparative genomics under 4 heads. The first head is what I call when BLAST tells you nothing. The next head is the guilt by association principle. Then we'll introduce you to 2-dimensional gene annotation, functional annotation, and the subsystem concept as it exists in the seed database that you'll be using, which is based on 2-dimensional annotation. Then we're going to look at 2 plant prokaryote examples of using comparative genomics to do the 2 classic operations that comparative genomics does, which is to fill pathway holes, which is to find missing genes where you know the function exists, you just haven't yet found the gene for it. And the other example is where you have a gene, all right, but you don't know its function, so you go through processes that allow you to link that function to some known system. So the first thing is we have a function but no gene for it. The second one, we have a gene but no function for it. And those are the 2 basic cases. That's the basically hour-and-a-half lecturing I'll be doing. Then we'll briefly learn and do some exercises on the Stream Comparative Genomics database, or website database and tools. This is a very good place to start a comparative genomics project. It's totally automated. There's nothing you can change about it, but it's very rich and it's getting richer. It's a great place to learn about. We won't spend very long on that. It's very intuitive. Once you know how to use it, you can just follow logic. Very briefly, we'll touch on a few other web resources, but we're going to spend the bulk of the time introducing you to the seed database and its tools and related tools for comparative genomics. And this is a very powerful database and the tools, but it requires significant instruction before you can fly it. And that's what you're going to have in the first, particularly the first day and tomorrow somewhat. But you can only really learn to fly it by taking the controls, and that's what you're going to do with building subsystems. Some of you brought your own problems, others will give you problems to work on, and that's the way of familiarizing yourself with how to actually exploit it to its max. Firefox is definitely authorized. Sveta, what else would you endorse at this point?
Safari works, but worse.
Definitely not the Net Explorer. Just sort of introduction to this missing or unknown gene, the scale of the unknown gene problem. If you look at progress in sequencing whole genomes since the first genome 20 years ago, almost, that's what it looks like. The blue bars are complete genomes, the yellow ones are the ongoing genome projects. That's the number of genomes. By 2011, which is the last date I think people have been taking these statistics, we had 10,000 genomes. I looked last night, we're up now to over 27,000. This is only going to increase because of next-gen sequencing of genomes. So we have enormous numbers of genomes now. Increasingly, they're from eukaryotes as well as prokaryotes. But functional annotation of the genes in those genomes, in other words, ascribing a well-defined function to the genes, has nothing like kept pace with the speed at which the genomes have been sequenced and put out in the public sphere. So the functional annotations are often absent altogether. You've probably all seen hypothetical protein as an annotation. Yeah, well, actually, that's a good annotation. There's a lot ones worse than that. It's where they're wrong, because that tells you that it's known when it isn't. So sometimes they're absent, sometimes they're wrong, and sometimes they're just vague, and that would be GTPase, esterase, oxidoreductase. Tells you the family, but not the function. So this gives rise to a gigantic problem that's been called orphan genes, in which roughly 20 to 60% of the genes in any given genome have no known function, or at best a vague one like esterase. So if you kind of multiply that by the number of genomes, it means that you have more than a million genes that belong to thousands of different gene families that have no associated function in published genomes. So this is a huge problem. And there's a reciprocal problem, not so well recognized. It's orphan enzymes. And as a biochemist, I was shocked when I first saw these statistics. There's about close to 4,000 enzymes have an EC, an enzyme commission number, but more than a third of them at this point in history still don't have a gene to go with them. So the biochemistry's been done, but no gene has been hooked to it. And the problem is actually a bit bigger than that, because only about 2/3 of known enzymes have actually been classified yet. So if you put those two numbers together, you can come up with an estimated about 2,000 orphan enzymes that are out there that are known to science, often have extensive publication documentation, but we don't know the gene. Now, the unknown protein problem, the things in genomes that encode proteins and we don't know what they do, it's getting worse, because this is a plot of the growth in the percentage of domains of unknown function in the PFAM database, sequence classification databases. So in PFAM, things which we don't have an unknown function are helpfully called DUFs, domains of unknown function.
It's something I learned I didn't know. The lower the number, the more genomes have this domain of unknown function. So DUF1 would be present in every genome, whereas DUF 1 million means it's just present in one or two. So this is something I actually learned not long ago.
So here's the percentage of domains of unknown function as a function of the number of PFAM families that have been recognized. So basically, this is a kind of time axis. The more genomes you sequence, the more you recognize that there are families of proteins, and you classify them, and PFAM's now up to 10,000 families classified. But the more genomes you sequence, the more families you find, the more of those get to be unknowns. So, in other words, ignorance is increasing with time. It's kind of counterintuitive. Maybe what's even more counterintuitive is that this problem of orphan enzymes is getting worse. Because if you plot the growth In total EC numbers, in other words, that's the total enzyme commission enzymes that have been classified in that system. If you plot the growth in EC numbers and the growth in ones that haven't got an identity, that's the cumulative EC numbers since the project was started in the early 50s, and it's, comfortingly, it's basically a straight line. It says biochemistry isn't dead. We're still discovering things. But look at the plot for orphans. I mean, the number of orphans is still going up, so we're still actually discovering enzymes that aren't being hooked to genes. So this brief overview is to give you a kind of visceral feel for how much is to be discovered out there, both in terms of functions for all the genes that we don't know and genes for all the functions that we do know but don't have a gene. I told you at the beginning, hypothetical protein is in a sense a good annotation because it's honest. It says we don't know what it is. A much worse annotation is one which is wrong, or what they call over-annotation, which is more specific than it should be. And there's a very nice study of this, of misannotations, done by Babbitt's group a couple of years ago, where they took 37 very well characterized enzyme families and looked at the change in misannotation over time in GenBank. So that's the number of sequences, and the block. Ignore the dots connected; they're not really helpful. In 1993, say about 80 sequences were put in. The green part of that is correct, tiny little incorrect part, and that carried on till the end of the 90s. After that, genome sequencing really came in, and people started propagating annotations by homology, as opposed to actually knowing what the gene did when they put it into the database. And as you can see, as the number of sequences has gone up because of the sequencing of genomes and the propagation of annotation by homology, more and more of them prove to be inaccurate. So you're up to about 40% now are in this even this very well-known family are wrong. So beware in NCBI, because this is specifically NCBI data, a very high proportion of, even in well-annotated families, are misannotations. And this is a serious problem because it degrades the value of the information in the database. There's two kinds of phenomena going on here. Many unknowns are misannotated as knowns, and vice versa. And the biggest problem is what it says in the heading there, misannotation. It's over-annotation. In other words, you give something a spuriously specific annotation. You say, all it really is justified in being called is, for example, an NAD-dependent dehydrogenase. But then, by homology, somebody will have decided, or a computer will have decided, that it should be called some specific sterol dehydrogenase. There's no basis for that. It's just a guess. And so something will have an extremely specific annotation. They're treacherous. And some of them are laughable. I mean, you'll be looking at a bacterial genome and it'll tell you it's a mitochondrial precursor from a mammalian drug resistance gene. Ding! That one must be wrong. But most of them are not that easy to spot. Now, this problem of unknown proteins, in other words, genes that encode proteins whose functions are unknown, it's a problem in all the groups of organisms, but it's somewhat worse in eukaryotes. And here's a plot showing you a very, very generous interpretation of what's a known gene versus unknown gene in different organisms, starting with bacteria and the best known genome of all, E. coli. A very generous estimate would be that we know 80% of the genes in E. coli. A more realistic one, it would probably be about 70, although it's a sort of chilling fact that only 51% of the genes in E. coli actually have experimental evidence hooking the function to the gene. So it's a pretty small genome, 4,500 genes, best known organism on the planet, still experimental evidence only for half the genes, and we've got about more than 1,000 genes in that genome that we don't know what they do. As you go to larger bacterial genomes and more exotic lifestyles, you get an increasingly large percentage of unknown genes. When you move to archaea, it gets larger still, and when you come to eukaryotes, it's larger yet again. These are, again, very generous estimates of what's known in Arabidopsis. I would put it nearer to 20, if you know, by a criteria that I would be happy with. Huge problem. So, I've just been giving you a whole bunch of bad news. This is where the good news starts. Plants, and that there could really say eukaryotes as well, and prokaryotes share a lot of genes, including many of the unknown ones. And specifically for plants, there have been various estimates made for the model plant Arabidopsis of how many genes in Arabidopsis are shared with prokaryotes, but all the estimates agree that there are many thousands. And an important point is that most of the shared genes between eukaryotes and prokaryotes are metabolic in the broad sense of the word. That would include, you know, replication, transcription, translation, DNA replication, as well as small molecule metabolism. And there's a reason for that. It's that the regulatory mechanisms in eukaryotes and prokaryotes are very different. They don't share those. The regulatory proteins are different, and the structural proteins tend to be different as well. So all you're left with is metabolic, in which I include transporters, by the way. To me, those are enzymes. And if you look, we made an estimate, Valerie and I, about 6 years ago. It's quite like other people's. We thought that about a total of around 11,000 genes out of the 26,000 total in Arabidopsis are shared with prokaryotes. That's more than 40% of the genome. More than that, the genes are identifiably from various groups. In other words, the closest homologs of the plant genes that you find in prokaryotes will be from a specific group. And as you might imagine, cyanobacteria lead the field. You know, 20% of plant genes that are shared with cyanobacteria. Yeah, symbiotic origin of the chloroplast. Yeah, it's come in and imported the genes with it, and then they've subsequently migrated out into the nucleus in most cases. Another important group, proteobacteria, especially alpha proteobacteria. Why that makes some kind of sense? Yeah, the origin of mitochondria in alpha proteobacteria. Then there's some mysteries that we really don't understand. Gram-positives, a lot of those. Actinobacteria and other bacteria, no rationale to explain that. And archaea, quite a lot. Why archaea is logical. The sort of the cell that imbibed the mitochondria and the plastid was a kind of archaeon. That's the theory. So particularly this applies to the core transcription translation machinery. So you could think of plants or other eukaryotes as being conglomerates of microbial metabolic genes. That's one way to look at it. So that gives you many opportunities for doing comparative genomics of these unknown proteins or looking for candidates for functions that we know, because you've got this wonderful shared overlapping gene families between plants, animals, prokaryotes, fungi. A little digression on why plants and prokaryotes share many genes, and this comes from a really wonderful paper by David and Alm a couple of years ago. It gives you a big picture evolutionary rationale. What they did was plot a time axis from the present going back 3.5 or more billion years. So this is a time axis. And they plotted their estimates of the gene loss rate and the gene gain rate with 0 there. And they did that for the birth or appearance, you know, from nothing of new gene families, for the transfer of new gene families, for their duplication, and for their loss. And when you look at this picture, there's this huge explosion of the invention of new genes about 3.2 billion years ago. After that, the rate of invention of new genes really falls off. And the processes that take over are transfer, duplication, and loss has been going on all the time. Now, on that time axis, look where the very first true plants appear 1.5 billion years ago. The creation of new genes is almost already nothing. By the time you get to land plants and even more angiosperms, it really is nothing. So it's true that you get, particularly regulatory genes get invented in eukaryotes. But they're a comparatively small fraction of the total genes if you look at the whole of biology. And so what's really happening in plants is that they're dealing with a set of gene families and kind of generic functions that were invented 3 billion years ago, and they're just shuffling them. They're not creating anything de novo, so that means that there's a high probability that they're going to be sharing some of those genes with other organisms, because they've all inherited them from way, way back in the tree of life. The power of comparative genomics. This may be a familiar experience to some of you. Suppose you have an unknown eukaryotic protein. You don't know what it does. You do a BLAST-P search against the non-redundant database, and you find it has various hits on prokaryotes, good ones. You get encouraged. And then you read the annotations, and none of those prokaryote hits have clear functions. You could perfectly well substitute unknown eukaryote protein. You could substitute unknown prokaryote protein there as well. So you've reached a dead end. Well, no, you haven't. This is where comparative genomics begins. Because comparative genomics predicts functions by the guilt by association principle. And different cultures have different ways of expressing that principle. In English, we say, birds of a feather flock together, or show me who your friends are, I'll show you who you are. The idea is who you hang around with is a pretty good guide to what you are. And genes of related function can be associated in various ways. And when we say genes of related function, we mean, for example, the enzymes in a pathway or the proteins that belong to a molecular machine, a complex. And just like the saying, show me who your friends are, I'll show you who you are, whatever a gene's associates do, it probably does something in that same area itself. So that's the whole basis for comparative genomics. There are various kinds of association evidence. The two big categories are genomic, which means they come purely from genomes. The other category is post-genomic. It's necessary that genomes be there, but it's not genomic information itself. In genomic evidence, the most, you could say, common and the most powerful is gene clustering. In bacteria, archaea, genes are commonly arranged in operons, which are translated together, and the contents of an operon are very frequently functionally related. An important thing is that genes in prokaryotes don't actually have to be in operons. They can be just clustered together, and they can still be a strong functional link. Just the fact of being nearby on the chromosome is already a very strong indication of possible similar functions or related functions. They don't actually have to be in operons. But let's take an operon as a simple case. If we know what A, B, and D do, it's a fair bet that C does something related to A, B, and D. Even stronger, unfortunately not as common, but even stronger evidence for function are where you get genes fused. where you get open reading frame ORF-X and ORF-Y in some organisms are separate, in others they're fused. Well, if we know what ORF-X does, it's a very fair bet if sometimes it's fused to ORF-Y that it's in some way the function of ORF-Y is related to that of ORF-X. Another kind of evidence that we're going to be dealing with both of those in our class. We're not going to deal with these shared regulatory sites, but as you probably know, in not only in prokaryotes but also eukaryotes, genes that are spatially separate in the genome, they're not anywhere near each other, can be preceded by common regulatory sequences that are the same, and that they bring about co-regulation. And that could be the binding sites, the DNA recognition sites, but for transcription factors. In bacteria, it could be riboswitches. So these regulatory elements can tie together a group of genes functionally. And again, if we know in a series of genes that have the same regulatory element, we know what A, B, and D do. We don't know what C does, but we do know it has the same regulatory element as those. Very strong argument that C is functionally related to A, B, and D. And lastly, in purely genome-based evidence, all you need for this evidence is genome sequences. Lastly, you have phylogenetic occurrence, where, let's say we are thinking of a pathway, a metabolic pathway, which is absent. This is meant to be a schematic tree of phylogenetic tree. The pathway is absent in certain organisms and present in others. It should follow that all the genes for the pathway will be present in this organism and all the genes will be absent in this organism. So if you track the distribution of genes between organisms, if two genes have a very similar distribution pattern, that might be because they are components of the same pathway. So this co-distribution, very helpful too. But we'll see some examples of this when we do our exercises, and it's pretty clear when you see a case of it. So that's what you can get out of genomes in themselves. Post-genomic evidence, which depends on there having been a genome, which is then used as a resource to build other resources, that can also be very useful. You have co-expression. This is meant to be like a microarray output summary. In eukaryotes, that really takes the place of gene clustering, because clustering on the chromosome isn't completely clustering of functionally related genes on the chromosome isn't completely absent in eukaryotes, but for all practical purposes it is absent. It's common in prokaryotes. I think the estimate's about 40% of prokaryote genes are probably clustered together in some functional way. Co-expression, expression of the gene in the same place, in the same organ at the same time, that kind of replaces clustering for eukaryotes, because very frequently that's exactly what happens. A gene will be on in kidney but not brain or liver, or it'll be on in root tips but not in leaves or fruits. Co-expression, genes that are expressed together could have the same function, similar functions. You have now wonderful protein-protein interaction databases, very noisy, a lot of spurious numbers in there, but sometimes they're useful. For plants and animals alike, but especially plants, we now have organelle proteomes, not only predicted bioinformatically, but determined by mass spec analysis. And for chloroplasts, for vacuoles, for mitochondria, now even for the cytosol, these are sometimes very useful to help you reject a hypothesis. Because if you're looking for a missing gene in a pathway, which you know is mitochondrial, and your predicted gene, or maybe experimentally established gene location, is in the chloroplast or the vacuole, it's very unlikely that that's a good candidate. So you can use this as a sieve to throw out candidates which are maybe not promising. We also have now, for bacteria and growing increasingly for plants, we have essentiality and so-called phenome data. Phenome is just a word that means high throughput phenotyping. So if you find that your gene of interest, when knocked out in a given bacterium, causes lack of growth, that means it's essential, that makes it interesting. And in plants, if you knock it out and it goes purple or it goes small, that's interesting too. And in itself, that may give you a clue to function. And then last of all, we have structures which can be very helpful. Firstly, to classify a protein when it may not be possible to do that just from the primary sequence. At the very best, structures contain bound small molecules that are substrates, products, or related to substrates and products, and those can be really helpful clues to what the function is. It's not common, but it happens enough to be very valuable. So what you do with all these different kinds of evidence is you make predictions about functions. And when you've made the predictions, then you can apply the tools of biochemistry and genetics to test the ideas. And that's, again, something that we should emphasize here, that this workshop is not primarily for bioinformaticians, people who operate in silico. This is for people who are really more driven by the experimental facet of this work, but it's more important that it's very important that they learn to make the predictions themselves using this kind of evidence, because you as an investigator, a biologist with a problem, you have deep, sometimes you could say intuitive knowledge about function that may enable you to make much better predictions than a generically trained bioinformaticist, and even better than a machine. So we have a kind of a mission to make it possible for experimentalists to use some of these powerful tools to make their own predictions and not rely on machines or bioinformatician colleagues who are not experts. And as a experimental biochemist for 30 years before I met this kind of possibility of doing biochemistry by doing comparative genomics before you do the biochemistry, I found it to be enormously powerful, and I really do have a kind of a missionary zeal about teaching colleagues how to do this, because it opens up new vistas. And I mean, to put it in a nutshell, bioinformatics is too important to leave to bioinformaticians. It should be the experimentalists should have that skill as well. Conceptually, the dimensions in a two-dimensional gene annotation are the molecular function, of which a very precise example would be actually an EC number for an enzyme, which defines specifically the type of enzyme and the catalytic reaction it performs. That would be a very exact molecular function. The other dimension is functional context. In other words, we have an enzyme with an EC number here, a specific reaction, but does that make any sense? It's in a pathway. Is the enzyme before it in the pathway and the enzyme after it in the pathway actually in that genome? And if the answer is no, it might give you some question as to whether or not the EC number was correct, because something that's sitting there in isolation without all the rest of the infrastructure it needs to work is probably not a correct annotation. So I coined this aphorism. How many people have read Animal Farm? Okay, remember? 4 legs good, 2 legs bad. 2 dimensions good, 1 dimension bad. Because even an EC number function can be wrong if the pathway that that enzyme belongs to isn't in the organism, isn't encoded by the genome in which it occurs. And the pathway context can be wrong if some of its enzymes are missing. So remember, GenBank annotations are one-dimensional. They're just molecular function assigned in nearly all cases numerically by homology. Somebody dumps a bacterial genome in GenBank, you scan it for the closest homologs in some other organism and slap a name on it. And there's some kind of a cutoff on that homology search, and it's quite arbitrary, and so things which are not really the right function are given a function because it was somewhat homologous to an enzyme that did have that function. But now we come to the next stage in the process. We've just slapped a homology function on a gene that wasn't right. Well, now that's in the pool, And that's going to be propagated to something else by homology. So it's like the whispering game, you know, where you sit in a circle and somebody whispers President Lincoln in the ear, and it comes around the circle and it comes out a banana on toast. You know, it's complete distortion because you're propagating, and each step of the way there are mistakes. So that's why you should regard GenBank annotations as really suspect, because they're only done by homology and they only refer to molecular function. They don't ask the question, does this annotation make any sense in this genome? Now, when you're doing two-dimensional gene annotation, and I'm insisting on this because this is the power of the seed, this is one of its brilliant insights as to how to do the job of functional annotation better, the dimensions can vary in precision. A molecular function can vary from being extremely specific to general to unknown. That would be EC number, it's a very specific one. A functional context can be really defined, it can be ambiguous, or it can be undefined. And you can plot a kind of matrix in which you plot molecular function here from high to low precision, with a specific molecular function at the highest level with an EC number, a general function like esterase or GTPase or oxidoreductase, and then unknown. And similarly, for the dimension of functional context from high to low, you could have a defined pathway and have a general area of metabolism, which is ambiguous, or you can have an undefined situation. And this 2D matrix of the two dimensions of gene annotation defines the targets for comparative genomics. Now, in that matrix, which square or squares contain genes of truly known function, green, green? That's the only place where you really know that the function is true, because it's a specific E number and it fits in a pathway which is present in the organism. The context is sensible. So that and only that is your known gene. Now, in this case, you have a esterase that's associated with a defined pathway. Well, maybe this is a missing step in that pathway. And the same thing is true here. If you have a specific EC number associated in a general way with an area of metabolism, could this be an alternative pathway, or is it wrongly annotated? Similarly there, maybe you're dealing with a new pathway, maybe you're dealing with new pathways or missing pathways. When too much is unknown or vague, you don't have any chance. You're at sea. The best opportunities for comparative genomics tend to be where one thing is well defined, like a known pathway, and something is intermediately defined. So here and here are the two places where you tend to have the most power, because something is pretty well defined, something else is not. When the more vague everything gets, it becomes kind of combinatorial. There's too many possibilities. And the seed database uniquely implements 2D annotation. That's its power, that's why it works so well. And I want to introduce you here to the seed database, and especially its creator, Ross Overbeek, who created this, I think about, is it 15 years ago now?
Many incarnations altogether, maybe 20.
Yeah.
He was trying to, materialize his idea.
So when the first few genomes came out, Ross Overbeek, who's a computational scientist, had a stunning vision for somebody who wasn't a biologist of how best to make use of this information. And here is Ross, still very much active. And over the years, Sveta said, there were incarnations of the seed. It started out as WIT, and then it became Ergo.
Puma, Ergo.
And now it's seed.
Yeah, four.
Through private and public sector iterations. And I don't know, which iteration was this? Was this Ergo?
Yes.
So this is one of its iterations where it had really began to gather momentum. And you see Ross gathered around him a team of people, some of whom are very well known to us. This is Andre Osterman here, who's a collaborator, both Valerie and me. Here in the front is Sveta. And here are some other people. Veronica is still very much working with Ross. And
software developers are standing and annotators are sitting, but not.
No.
Rob Edwards is also doing a lot of stuff in SDSU. For those in California, he's in. No, sorry, that's the wrong Edwards.
So this is a remarkable story. For myself, I put Ross in the same category as what was the guy who did the human genome?
Venter.
Craig Venter. I mean, they were contemporaries and they both had visions that the rest of the field didn't have. And it's been a real privilege to get to know Ross a little bit and to work with the seed. This really is somebody outstanding, and we have all to thank him. The subsystem concept that he implemented, subsystems in the seed capture both these annotation dimensions, the context and the molecular function. And they are just sets of molecular functions, like enzymes or transporters, that together implement a specific biological process like a pathway. So it's a group of functionally related entities, and they cover many genomes, and they have the form of a spreadsheet in which the columns are molecular functions, the rows are genomes, and each cell in the spreadsheet identifies the genes for proteins with the specific molecular function in that genome. Now, this will make sense when I show you what a subsystem looks like, and in our exercises you're going to build one of these, a small one, as a practice with us, and then hopefully you will build your own surrounding or about a problem of interest to you. So here's what a spreadsheet looks like. This is a very small spreadsheet in one sense, because the genomes, the rows, there's only a dozen genomes in here, less than a dozen. A real spreadsheet could have 3,000 rows in it, because that's the kind of numbers of genomes that are in the seed now. So I'm just showing you a very small subset, if you like, of the whole thing, which would go up and down. It would go up to the Himalayas, and it would go down to Australia if it was the real thing. And the columns here are the enzymes in folate biosynthesis pathway, the vitamin folate. So your columns are the so-called molecular functions, the enzymes, and the rows are the genome. Now, the first thing I want you to look at is this set of 3 Bartonella genomes here. Remember that when you see a number, these are actually links in the cells. This means that there is a gene in that organism corresponding to this function, and that's the number of the gene in the organism. They're numbered basically, you know, from 1 to n,000 around the chromosome, if it's a prokaryote, or from, according to some eukaryote number system, if it's a eukaryote. So it means here we have gene number 458 in this genome encodes FolE, which is the first enzyme of the folate biosynthesis pathway. When you see multiple entries in the column, it means that there are 3 copies of that gene in the chromosome, and you can look at the numbers, 1800, 3742, they're obviously quite far apart on the genome. So let's go back and look at these Bartonella genomes. What can you tell me by looking at that pattern? Well, what's the first thing that strikes you about Bartonellas compared to the others? There's a lot of gaps, yeah, and a lot of genes are missing. Well, when you look at these with some detail, what you see are missing from this folate biosynthesis pathway of almost all the genes required for de novo synthesis of folate. The only thing you've got are what you need to salvage folate from the environment, which are the genes we have, you know, because we rely on a dietary supply. We've got those genes. So you can just look at this and say this organism requires folate. It doesn't have, it's not got just one missing gene, it's got the whole lot missing. So there's no way can this make folate. This isn't a trivial problem of a gene that we haven't somehow found. All of them are not there. So that's one of the ways that genomes can be used. It can be used to predict, to make a metabolic model of the capabilities of the organism whose genome you're talking about, because you can look at it and see what can it do, what can it not do, what does it require from the environment, what could it potentially supply to another organism with which it was living. So it's a very much more holistic, almost ecological kind of thing you can do with genome information. You can predict what the organism is capable of doing and to what extent it depends on other organisms for components of its biochemistry. So that's a case of a very incomplete pathway. Let's look at that one now. That's an enzyme of folate biosynthesis. What can you tell me about that? Well, that's actually right. It's simply a nothing gene in everywhere. So we know that gene exists from biochemistry. We've even given it a name here. But at the time this spreadsheet was constructed, that gene wasn't known in any organism. It was what we call that a universally missing gene, or a pathway hole. That's another informal term for it. And we're actually going to, this gene is now known, we're going to show you how it was discovered. We're going to reconstruct the comparative genomics of how it was found, which was very straightforward, in a minute. So we're now just going to close with some plant prokaryote examples of comparative genomics, the two kinds I mentioned at the start, which are the case where you have a known function but no gene for it, and the case where you have a gene but you don't yet have a function. But before we do that, just a few brief comments. From prokaryotes, the association evidence is mainly genomic. Clustering, fusion, co-regulation, co-distribution. Whereas for plants, because gene clustering in plants is almost always not important in terms of function, the association is mainly post-genomic. Post-genomic evidence is noisier than genomic evidence, but it's still very useful. And there are superb plant post-genomic resources. You have very good microarrays and increasingly RNA-seq data sets for specific organs and specific environments. You have very good organella targeting prediction and now proteomic. And as we've said, location can sometimes be used to rule out functions, hypothetical functions. We have beginnings of phenome databases for plants where you have systematic knockouts of all the genes and then report the phenotypes. And we have huge EST databases and of course a vast literature on plant metabolism and plant chemistry. Okay, so now we're going to reconstruct, and we're going to do this again when we come to the first exercise with string. We're going to go through this, how to find a pathway hole, the case where you know the function, you just don't have the gene yet. Well, this case we've drawn the folate synthesis pathway where we saw those were the columns in the spreadsheet I just showed you. We've drawn it out as a sequence of reactions. You can see there are genes known for all the specific steps in that pathway, except a few years ago this fold Q was missing. This is a pyrophosphatase that takes a triphosphate derivative of dihydroneoxyrin DHN and turns it into the monophosphate. So it's a fairly typical pyrophosphatase type reaction. As we said, it's universally missing. The organisms that make folates, which include a lot of bacteria, all plants, fungi, some protists, no gene known in any of them. The missing step was known to be a pyrophosphatase because we knew what the reaction was, and that enzyme had actually been purified in some wonderful classical biochemistry in the 60s by Gene Brown's lab, and we knew it was a small pyrophosphohydrolase, about 17 kilodaltons in E. coli. So all it was necessary to do was to search genomes for genes encoding a small hydrolase, preferably a pyrophosphohydrolase type hydrolase, that clustered with other fold genes, in other words, known genes of folate synthesis. Because we're using the argument, genes are clustered, we're missing a gene, the gene that's missing will be clustered with other folate synthesis genes. Those are the steps in the argument. Well, one candidate jumps out right away. It's in firmicutes, particularly in Lactococcus, but in others too. it's called YLGG, is the systematic gene name in Lactococcus. A brief digression about prokaryote gene names. When genes have known functions in prokaryotes, they're given a name that's some kind of acronym related to that function. Until then, genes are given in prokaryotes and yeast, they're given systematic names which are like this. Valerie, systematic names for unknown genes in bacteria always begin with Y?
No, the Y system was just developed for E. subtilis and E. coli. For the other genomes, they use locus tag that starts with a CT, like the organism abbreviation. So only E. coli and yeast, and E. coli, yeast, and B. subtilis had the Y. Salmonella, not really. They just hooked up on the E. coli one.
Okay. Yeah, so this is a systematic gene name. Doesn't mean we know the function. It's just been named with a code. It's a member of the Nudix hydrolase family. It encodes a protein of about 19 kilodaltons, and it's sitting right in the middle of a folate gene cluster. So here's a folate gene operon in Lactococcus lactis. That's a fusion gene between two of the proteins. That's FOLP, that's FOLC, and sitting between them is this YLGG. That was our candidate because it fit all the description. It's clustered with folate genes. It's a small protein. It's a member of a pyrophosphatase family. And actually it has a plant homologue. Not a very strong one, but it does have a plant homologue. So we're experimentalists, so we made experimental tests. And it's not difficult to see the sort of tests you do. You'd predict that if you knock that gene out in Lactococcus, you get an accumulation of that, and you get little or no folate. Well, that's more or less what happens. If you knock it out, these are HPLC traces of the wild type and the knockout. That's the peak of this compound in the wild type. This is the peak in the knockout. You can see a bunch of other peaks. These actually include the monophosphate, because what happens is when this accumulates, non-specific phosphatases kind of take over, and you get a kind of messy reaction that gives you a little bit of the product. So this mutation is actually not non-lethal. The cells are depleted in folate, but they're still alive because it can kind of make it through with non-specific reactions. And when we test the recombinant proteins for activity on this substrate, they have nice activity. They cleave this into the monophosphate, this product, and pyrophosphate, not inorganic phosphate, and the same is true of the plant homologue. So this is a very classical story, very simple one. It was actually my introduction to comparative genomics. This was a gene that had been missing forever, and we found it very easily, actually. This is a more challenging case where you have a gene, all right, but you don't know what it does. And I'll just show you right up front what it does, or what we now know it does. It's a folate-dependent protein that's involved in the repair of iron-sulfur cluster cofactors, especially in oxidative stress, because iron-sulfur cofactors are very sensitive to oxidative stress. We've given it a name. It's this from this COG database that we talked about earlier on. It's a systematic name. It's a name for a protein family. It's not a name associated with a function. It just refers to a group of proteins that have sequence similarity. Let's just look at the things you can find out about this protein by a blast search first. Well, you can find that it's in all kingdoms of life. Here's a family tree, a phylogenetic tree. It's in bacteria, it's in some archaea, it's in fungi represented by yeast, it's in animals and protists, and it's in plants. And you notice right there that there have been two plant genes have flown in, and the first one at the top is related to rickettsias, which are alpha proteobacteria. You see it's phylogenetically coming out with the rickettsias. These are the closest living relatives of the progenitor of the mitochondrion. And the other one is clustering with cyanobacteria, and of course those are the progenitors, or these are the living relatives of the progenitor of the plastid. So that's a nice story, and you actually see that not uncommonly. There are often, when there are two genes in plants, one of them may have come from one ancestor, one may have come from another. This gene is often annotated in databases, and this is a good example of the problem we talked about of over-annotation or misannotation. It's annotated as GCVT, which is the folate-dependent glycine cleavage protein from the glycine cleavage complex. But it's clearly a distinct clade to that, because if you just do a phylogeny of GCVT proteins from yeast, mouse, plants, E. coli, it's obviously a separate clade. This isn't the same thing. You don't even have to do comparative genomics to see this, but this is annotated all over the place. This family is annotated all over the place as being GCVT. Typical. Even though in the genomes in which it's annotated as GCVT, there's a much better candidate, which is a better homologue to the known GCVTs, which is usually clustered with other genes of the complex. So a little bit of context information tells you right away, actually, that this is probably not, but so does this phylogeny. And these one-dimensional databases like NCBI are blind to the fact that it doesn't fit.
Yeah, and over-annotation of paralogs, which is a case you have here, is a very, very common problem. So a flag to a problem in an annotation is that you have paralogs. That should be, oh my God, I have to double check.
So just to back that up, you see, this is E. coli GCVT. This is the E. coli COG034. That's the Arabidopsis GCVT, and these two are Arabidopsis COG034s. So this group diverged way before the speciation here. And it's actually, thank goodness, it's very easy to tell this apart from GCVT because it's got a very characteristic motif that you can just use to say right away, is this a GCVT or is it a COG034? That much you could all tell by blast searching and a little bit of phylogenetic tree construction. Now let's go to what you can learn from comparative genomics. All you can really learn from BLAST is, yes, it's all over the place. It's an obviously very ancient protein. And no, it isn't GCVT because it doesn't phylogenetically group with it. But let's look at comparative genomics now. What can that tell you? Well, let's look at co-expression in Arabidopsis. You find that the mitochondrial version of the protein correlates in expression with frataxin, which is an iron sulfur assembly protein, and you find that it also correlates somewhat in expression with ferritin, which is an iron storage protein. So we've got an iron sulfur connection here from just the plant information. That gets a lot stronger when you start to look at the prokaryote members of this protein family, because they cluster over and over again with different proteins that have iron sulfur cofactors. And I've just put a sampling in this slide. The COG035-4, our mystery gene, is in blue. The iron sulfur proteins themselves are in bright red, and what I'd call iron sulfur partners, which are members of a complex with the iron sulfur protein or members of the same pathway, are in rows. As a typical example, just look at this one, an NAD synthesis cluster in Pelagibacter. There's our COG035-4. It's in an operon or an operonic structure with NADA, which is an NAD synthesis gene which has an iron sulfur cluster, and NADC, which is a partner of that. Even simpler cluster here, MIR-B, in quite a few organisms, has an O354, COG035-4 with it. This is a radical SAM enzyme, which means it has an iron sulfur cluster. And then much more elaborate clusterings.
What database did you use for the previous point, the co-expression?
The co-expression? There's several of them that will do this, but the one I use, it's in the GOLM database in Germany. When we come to the other databases, resources, I'll mention that. Some other evidence you can get from prokaryotes is co-distribution evidence. It only occurs if the gene ISCA is present. Now, ISCA proteins are scaffolds involved in assembling iron sulfur clusters, so you don't find our protein unless there's an ISCA protein encoded by the genome. Here again is a family tree of prokaryotes. The red boxes are where our gene occurs. Those boxes are where ISCA occurs. You can see there's a few cases where there's an ISCA but not our gene, but there's never the reverse. So a precondition, if you like, is that there should be an ISK present before you have our gene of interest. So that's the first of actually 3 connections with ISK distribution. Our protein, if you look at the genomes in which it occurs, it's very common in aerobic organisms, very uncommon in anaerobic organisms, which starts to say connection with oxygen, and strengthening that connection, it's induced by oxidative stress in E. coli. And high throughput screens, essentiality and phenomics, tell us it's an essential gene in some bacteria, and luckily an important but not absolutely essential gene in E. coli, where if you knock it out you get slow growth, and in yeast you get a petite phenotype. Anybody know what a petite phenotype in yeast signifies? It means small colonies, yes. Beyond that, what does the small colony connote? Yeah, malfunctioning, a mitochondrial malfunction. These have actually lost their mitochondria. And high throughput proteomic screens show us that both plant proteins are expressed, that the cyanobacterial-like one is in plastids, and most importantly, modeling of the E. coli structure shows that the protein has a folate binding site similar to its relative protein GCVT. And synthetic genetic arrays, and these are projects in which you knock out pairs of genes, so you're looking for genes that become lethal if another gene is knocked out. So on its own, the mutation isn't lethal, but it becomes lethal if something else non-lethal is knocked out, and it's ISK genes. So that's our second connection with ISK. The first was distribution, now this synthetic lethal. Literature mining, that's an important part of comparative genomics. There's now computational tools that help you do that. We learn that plants make iron sulfur clusters in both mitochondria and plastids, so that would make sense for there to be 2 versions of this protein in plants in those organelles. We learn that the yeast protein complexes with escape proteins. Okay, that's our third hookup to ISK proteins. And we know that the E. coli mutant low in the E. coli mutant in our protein is low activity in MIA-B, which is an iron sulfur enzyme. So if you put all that together, we can make some predictions about this protein. We can predict that it's a folate-dependent enzyme, like its homologue. We can predict that it's combating oxidative stress, that it helps make or repair iron sulfur clusters, and that its function is ancient and ubiquitous like iron sulfur proteins themselves. There are even people who postulate that iron sulfur proteins came before life. They were part of the prebiotic world. Well, we can make experiments, and we can show experimentally that folate mutations abolish its activity. In other words, COG0354 doesn't work if you take away folates. That supports it being folate dependent. We can show that the mutant is oxidative stress sensitive, and I'll give you data for that in a moment. We can show that that mutant has many iron sulfur enzyme defects. Take it away, several iron sulfur proteins go bad. And you can, in keeping with its function being ancient and ubiquitous, you can complement the E. coli mutant by that gene taken from any one of the kingdoms of life, which is really striking. So here's the data. We're growing E. coli here. We're actually growing the mutant where we've knocked out this COG0354 gene, and we're growing it on an oxidative stress medium. So when we grow the mutant and only put a vector in it, nothing happens. It doesn't grow. When you replace the knocked out gene with the E. coli gene on a plasmid, these 2 controls, everything grows normally. That's what the wild type would look like if it were on there. If you replace the E. coli gene in the plasmid with the plant mitochondrial, the plant chloroplast, or the mammalian gene, everything still works fine, so you can complement with plant or animal genes, and you can do the same, not quite so well, with fungal, protist, and archaeal genes. So that's in a kind of very compact way, that's how you use the comparative genomic evidence, different kinds, blend them into a series of hypotheses, and then use bacterial genetics and, of course, also biochemistry, which I'm not showing you, to test whether this prediction is valid. So that's comparative genomics in a very brief span. Before we go to the databases, I want to leave you with this wonderful thought, which is so relevant to comparative genomics. And it comes from William Whewell, that's pronounced Whewell, who was basically a mid-19th century scientist, philosopher. He was also an Anglican priest, as many Oxford and Cambridge academics were in those days. And he was an early influence on Charles Darwin. He was one of Darwin's teachers. And he coined the term scientist in its modern use. So every time you use the word scientist, you're actually remembering William Whewell. And he said, the facts are known, but they are insulated and unconnected. The pearls are there, but they will not hang together until someone provides the string. And it's that providing the string, linking these different kinds of evidence together into a comparative genomic hypothesis, it's that hypothesis that connects and unifies observations that you're able to make with these online resources and web tools that go with them. And that's what we're trying to convey to you how you can do in this workshop.